We're into lab activity 11, and we're starting to look at histology of epithelial tissue. And this first sample here, we're looking at simple, meaning a single layer, squamous, the flat shape, and in this section, we're looking at epithelial tissue. And we get the full name there, simple squamous epithelium. On this particular sample, we're looking right here at this internal surface. And we're looking at the inside of the artery. And everywhere you see this very thin, pink, wavy region right here. That's where we're seeing the simple squamous epithelium. And you can see the little sketch here. We're looking at an artery. So we have this hollow tube. And the entire picture that we're looking at would be zoomed in, say, right about here where I'm drawing the box. And again, the inside surface of a blood vessel is one example of where you would see that simple squamous epithelial tissue. And we could go ahead and take a look at another image, zooming in closer. And we see just again how thin it is, simple one layer, the squamous, the flat, thin shaped epithelial cell. The next sample here, we're looking at simple cuboidal. And in this case, I'll make a little sketch again. You have to imagine we have a tubule. And now the entire tube itself is made of these cuboidal cells. So if I draw a bunch of little boxes, these are cuboidal cells. And you notice cuboidal cells have a rather large, centrally located circular nucleus. And that's what we see here. So I can highlight this section. And again, it's made of these individual little boxy shaped cuboidal cells that just happen to be brought around into a ring structure. So here's a sample for simple cuboidal epithelium. And you notice, of course, it's not everything that we see here on the image, but we have some good examples of simple cuboidal epithelium. And then another simple example, single layer. In this case, the basement membrane would be located here. Let's put BM, typically found just at the very deep edge of epithelial tissue. And now we could highlight the cell membrane. We'll see that these cells have much more length when compared to their width. So these long, slender cells, again, here they are adjacent side by side, but no stacking. So this is simple columnar epithelial tissue, simple columnar. So we had 3 examples of simple. Moving forward with the lab now, we have what's called pseudostratified ciliated columnar epithelium. So pseudostratified ciliated columnar epithelium. And of course, we would spell that out. So that's what we see right here. We could put a bracket on it, all of that PSCCE. So the pseudostratification is when it appears to have multiple layers. And here it definitely does look as if this nucleus is superficial to this nucleus. But truly, in pseudostratification, every cell does connect to the basement membrane. So there truly is only one layer. It's just that some cells are longer, taller, other ones are shorter. So pseudostratified, the cilia appear as these extensions here. And we'll zoom in closer. but certainly much longer than microvilli, the cilia. And pseudostratification is only ever seen with the columnar shape. Some columnar cells will grow longer, other columnar cells are shorter, and with the distribution of their nuclei, that's what now gives the appearance of the pseudostratified appearance. So putting it all together and zooming in, we have the pseudostratified, again, the cilia, the pseudostratified, ciliated, columnar, epithelial tissue. So that's everything that we're seeing right here in the bracket. And the cilia are a great indication of the pseudostratified appearance. So one time that we're going to see it. So now moving on to a stratified type. So multiple layers, at least 2 for stratification. So we have stratified squamous, those flat, thin cells, multiple layers of the squamous-shaped epithelial tissue. And now we need the distinction that this is the non-keratinized type. So that goes at the end of the name. Stratified squamous epithelium, it's non-keratinized. Or another way to say this is wet. Stratified squamous epithelium that's wet. And in life, that means in living tissue, this would be moist tissue. So for this example here, taken from the inside of the esophagus, that internal surface of the esophagus, and it's now here, the stratified, meaning that there's multiple layers, so we have that thick barrier now. Stratified, the flattened squamous shape, especially the closer we get to the surface, the flatter and more squamous like the cells are. And this is the non-keratinized or wet sample. And again, the full name, stratified squamous epithelium. And then we end with either non-keratinized or wet. As opposed to the opposite of wet, this is now the dry type. So stratified squamous epithelium. And this is the keratinized type, or just simply known as dry. And that means, of course, well, naturally in life, this would be hard, dry tissue. And this is what we see at the surface of our skin and our hair, for example. So the distinction is now, this is the very same type of tissue starting out with stratified squamous epithelium, but now it's the dry, hardened, keratinized type. So we still have that thick layer, it's stratified. Again, squamous, especially as the cells get closer to the surface, they become flatter and flatter. And the distinction to know that it's the dry sample is when you look right at the surface, and we start to see now the distinct layer of cells that are now shedding and attaching, just like it happens in our skin. So the desquamization, or simply the shedding and flaking you see of cells right from the surface, is the key that this is the keratinized or dry sample. And just going back to the non-keratinized or wet sample, we see that that superficial surface is quite smooth. And another look at the dry type, again, is where you see the flaking and shedding of cells. The wet sample does do this, but it's really bonded on. And you see that detachment. It has a very tight connection, and now it's slowly releasing. It is now a great indication of the dry type. This last one here is now transitional epithelium. So transitional epithelium. And for this sample, the basement membrane, I'll highlight its location. So again, along that deep edge of epithelial tissue, meaning that all of this darkened area here out to the surface, this is all the area of the transitional epithelium. And transitional epithelium truly is stratified. There are multiple layers. It's just not a part of the name. And the distinction now, how we could tell this apart, transitional epithelium, you notice as we get closer to the surface, the cells closer to the surface are actually larger and bigger in size. Whereas if we go back for the stratified squamous, dry or wet, we pointed out how the cells are thinner and flatter as they get closer to the surface. That's what we see on the stratified type of epithelium. Yet for transitional, we get these larger so-called umbrella cells that are closer to the surface. But it is truly a stratified type of epithelial tissue.